Okay, today I'm going to present a recent okay, today I'm going to present um background information of optogenetics. Optogenetics generally say is to linked with the light control and genetics and engineering. That it means that it can using the light to control the cell activity or response. We already know that our human eyes that can convert the light signal to electrical signal, then send back to the brain through the optical nerve. So this conversion is being done by a very thin tissue, the retina, that sit at the back of the eye. The retina tissues, here is a diagram of the shows that the light coming through the whole retina then hit the photoreceptors. On the outer segment of the photoreceptors, there are many discs. On the discs, there's a lot of molecules that are floating on the disc. It's called rhodopsin. Rhodopsin, this the name is coming from to combine two basic Greek words that is rhodo and opsin. Rhodo means this color like a rose, kind of a very pink color pigment, and opsin means the vision or see. So rhodopsin is like a pinky molecules that is a critical for us to see. So that's the name of rhodopsins that's coming from. Actually, rhodopsin is combined two parts. One is a protein that's called opsin, and another is like a bonding sit in the center of the opsin, and that's a retinol. For the human being, in the resting state, it's 11-cis retinol. So here we can see that a general um, process of a photo transducing is that at the dark and everything is at a resting state and a very high concentration of cyclin GMP and cyclin G channel that was gated by cyclin G is opened. So cation like potassium or calcium can flux into photoreceptor uh, uh, mm, inside of the cell. But if there's a light on or the light intensity is increased, then the 11-cis retinol will capture a photon and change to all-trans retinol. And the all-trans retinol will induce the whole rhodopsin conformation change, then binding to transducin. Transducin is a G protein and it will get rid of GDP then binding with a GTP, then dissociate with the beta GABA subunit of the transducin. Then the alpha subunit will bind into phosphorodiesterases, the PDE6. Then this binding will cause the active of the PDE6, then transform that hydrolyze the cyclin GMP to GMP. Then this step will reduce the concentration of the cyclin GMP. And the CNG channel is being closed, and the cation will stop to influx into the cell, and the whole photoreceptor's membrane potential will be decreased. So this is how this all this phototransduction process is going on in the photoreceptor. So again, it's a G protein coupled receptor that's a rhodopsin will activate transducin and it will activate phosphodiesterase, will decrease the cyclin GMP, and the channel cation channels are closed, and the membrane potential will be go more negative. And this requires a whole signal cascade and required a lot of molecules, channels, enzymes involved in this process. However, in green algae, the membrane potential change can be done by a single protein that is being called channel rhodopsin. After binding with the channel rhodopsin is just the same as the rhodopsin that they have a 7 transmembrane alpha helix and there's an all trans retinol that sits in the center of this this 7 transmembrane domain, and after captured a photon, then conformation will be changed from 11 cis retinol, from ultra retinol to 13 cis retinol. And this will cause a conformation change of the channel rhodopsin, and the channel will be open. And cations like proton, sodium, or calcium can flux through this channel. Here shows a structure analysis of the channel rhodopsin 2. And here we can see that they have a 7 transmembrane domain, and the N terminal is in an extracellular part, and the C terminal is an intracellular part. And all transretinal is sitting in the center, that is embedded in the membrane. From the top of view, we can see that the 7 transmembrane domain form like a barrel, and in the center, there's a cation goes through the channel, and the channel dopsin prefer that form a dimer, but the, the cations are flux into each. We can say that it's a subunit or a single channel rhodopsin that's in the center, not like some other research report that the cation may be flux between two channel rhodopsin. So far, scientists will prefer the first hypothesis that the channels are within each channel rhodopsins. So here is shows that it has a 7 transmembrane domain, and this color is being determined by the electrical polarity of each amino acid residues. For those that electrical neutral and is labeled with yellow, and the most have a negative charge or positive charge will be labeled with blue color. So here, this color do not mean that it's a negative charge or positive charge. It just means that it has a very electrical polarity. Either it's a positive or very negative. So we can also think about that like the oil is non-polarized and the water is a quite polarized molecule. So watercolor we consider it like a blue, and oil can consider like a yellow. So in this chart, like phenylene that with a benzene residue, that's the most like electrical neutral um amino acid, and arginine ARG is the most charged uh, um, amino acid residue. Then let's go back to see the structure of the channel rhodopsin, and we can see that close to the surface of the membrane, there's a more charge, and within the membrane that's sitting the insertion that into the membrane alpha helix are more like um electrical neutral um amino acid residues. Here is another knowledge point is that in a animal rhodopsin, it's the chromosome is 11 cis retinol, and after capture a photon, it will transformed to all trans retinol. And in the green algae, the cytine chromosome is the all trans retinol, and after capture a photon, it will change the conformation to 13 cis retinol. So that's a different. And it's very good to know that in the human being in the body fluid, less ab- abundant amount of the ultras retinal that's within in the body fluid. So there will be very sufficient supply of this ultras retinal to use if this channel rhodopsin is like it being expressed or transfected into like animal cells. So the first channel rhodopsin is being discovered like in 2002 or 2003, and after these 20 years, there are many several different kinds of like optogenetic tools that's been discovered from the microbiome. Um, like a chloride pump, like a proton pump, a sodium pump, proton pump pumping out that from intracellular to extracellular, or inward proton pump that pump the proton into the cell, and also cation channel and an anion channel at the GTACR that can like can open like and cause the chloride that go pass through this channel. And also in 2014 and 2017, there's a light activated sensor that's been discovered in fragile, and this either like light activate guanine cyclase or light activate PDE at phosphodiesterase. Um, the light activate phosphodiesterase and is image showing on a diagrams here that it combined of these two like transmembrane light light activated domain and the intracellular part of this phosphoesterase domain that can um hydrolyzed like cyclin AMP or cyclin GMP into GMP or AMP. And also like the human or mammalian rhodopsins are also being considered to be used as an optogenetics tools. So in this figure, we show that like some like a cation channel rhodopsin, and the inward proton pump can depolarize the cell, and the other pump like chloride pump or sodium pump, proton pump, or ion um anion channels like GTACR, a chloride channel, will cause the cell to hyperpolarization of the membrane potential. So this toolbox is a quite diversity, and there's a lot of things that can be chosen from this toolbox. And here is shows that the conformation change of this channel rhodopsin is not only like close, then after capture a photon, then they open. It's a still there's a different. There's several steps for a channels to be totally opened from a closed state to an open state. So the first step is a light-induced retinal isomerization that changes from the ultra retina to 13-cis retinal. Then the proton will be transferred from the Schiffer base, then bind into the Schiffer base, then cause small conformation change. Then the state from P1 to P2, then a helix opening for ion transporter in the CP site. That means that in the cytoplasma site and intracellular parts. Then this conformation change will change the state from P2 to P3, and the P3 is the name for a channel rhodopsin that's in the open state. Then it for the channel rhodopsin 2, it can automatically go back into the closest state, that's a thermal retinal isomerization. But however, like some other optogenetic tools, maybe you need to capture another photon to close the channel. So here is a background, a very simple introduction for the optogenetic toolbox. And thank you. Bye bye.